Hello, everybody. This is Jeanette Bruce. I am the program coordinator at Whistler Public Library, and I am coming to you live from Whistler Public Library. I'm delighted to be hosting tonight's Quest lecture, and I'd like to thank our partners, of course, the Squamish Public Library and the Pemberton and District Public Library. It was really exciting to be able to partner with our neighboring communities to offer these Quest lectures. And of course, I have to thank Quest University. Now, before we get into tonight's talk, I want to express my gratitude that we're able to gather here tonight on the shared traditional unceded territories of the Squamish Nation and the Lil'wat Nation. At least those are the territories that I'm on tonight. Maybe you're on a different territory. You can let us know in the comments. And I just want to express my gratitude that we are able to live, work, play, and learn on these lands. If you are watching on Facebook, I will be checking the Facebook comments for questions after Marina's talk. So please do feel free to ask questions on Facebook. If you're watching in Zoom, hello folks on Zoom. You can also ask questions in the Q&A panel or the chat panel. We should have plenty of time for questions at the end. And I am just going to tell you a little bit about our speaker tonight. So Marina Terlakis. Marina's teaching is focused on inspiring a lifelong love of learning and deeper engagement with the beautiful world around us. She aims to show that science is relevant to our everyday lives by using current research and popular media. She also incorporates her passions for drawing and science communication to help students engage with class material in non-traditional formats. So I'll leave it at that because you're gonna hear lots from Marina. We were just reminiscing about her past Quest lectures here in Whistler. One of them was on the subject of epigenetics, which I'm now quite proud to just throw into casual conversations. So Marina's giving me lots of street cred over here. But on that note, I am going to pass us over to Marina.
Okay, hello. Delighted to be here. Very different format from my previous library talk, but that's okay. So yeah, we're here to talk about viruses. Before we do that, I did also want to just take a moment to acknowledge that I currently am sitting in the traditional ancestral and unceded territory of the Squamish people, and to acknowledge that where I work, Quest University, sits on this beautiful traditional territory, and I'm very grateful to have the opportunity to work, learn, and live in this blessed territory. So for those of you who haven't been to Quest, you can see this striking image. You know, marketing always really does a good job of showing off, but it's quite easy, as all of us living on the West Coast know. Okay, so let's get into it. So viruses, that's what we're here to talk about today. And essentially, we're here to talk about viruses in a general context, and we will dabble in a couple of contexts, one of which is a particular interest of mine, and that is cancer, and the other, the thing that we all are constantly thinking and talking and worrying about, which is the pandemic. So just to give you an idea of what I was thinking about as I was organizing and planning for this talk, I had a couple of goals. One is the nerd goal. Well, I guess they're both nerd goals, but one is the primary nerd goal, which is to sort of share information and build appreciation for how cool viruses are, and to help everyone realize how much of an impact they have in our lives, both good and bad. And the other nerd goal that's in part particularly inspired by the current crises we find ourselves in, the climate crisis, the opioid crisis, and of course, our pandemic that we are all facing, and that is to empower audience members to consume science news critically. So I've thrown up a very short informal outline here, which is essentially keeping me honest in terms of trying to meet those goals. So we'll start off with me talking a little bit about the context and motive for this talk. which will then, of course, lead into talking about the subject of the talk, which is viruses and why they're neat. And we'll talk a little bit about how they work. And we'll talk, of course, about why they make us sick. And during the talk, I will bring up 2 recent examples of virus-human intersects. One, of course, will be COVID, and the other will be cancer therapies. At the end, I will take questions, and I will also perhaps pause at a couple of points in the talk and sort of do a weird eyebrow raise at Jeanette to see if maybe there are questions that I should answer right now that might be important for people before we move forward. Okay, so why am I giving a talk about viruses? So this is a bit of a slide that is meant to help me feel better about why I'm giving a talk on viruses. I'm sure many of you have heard about imposter syndrome, which is something that infects everybody. And so here's my caveat. I am actually not a virologist. I am a molecular geneticist, and I focus primarily on human disease, and I also have dabbled looking at plant disease. So why am I giving a talk about viruses? Because viruses have nevertheless been pretty prominent in all that I have done, from both my just education as a scientist in undergrad and through my training and now currently as I am an educator. So a very brief sort of tour through Marina's life as a scientist thus far. I started off in the lab of Dr. Katalin Hudak at York University, and that's in Toronto. And Kathy, which is how Dr. Hudak goes by, her main passion is something called the pokeweed plant, and specifically the pokeweed antiviral protein of the pokeweed plant. So in this little circle here, let's see, yes, I hope you can see my arrow. This is a picture of pokeweed. So it has these beautiful, whoopsies, little blue berries, and some people eat it, which is really quite puzzling to me because it's very good at killing cells. It does that because it actually kills, it dismantles a key component of a particularly crucial molecular machine, which I will get to in a minute. But as the name suggests, antiviral protein, this was probably the closest I've come to being directly involved in virology research, because my research was focused on trying to understand how does this protein confer antiviral abilities to this plant, and specifically, can we take the gene responsible and put it into another plant or an animal cell and help that cell become resistant to viruses as well, with agricultural applications as well as human disease applications. It happened that the human disease applications and learning about viruses especially was a trigger of this, while studying during my master's, made me want to make a shift. And so I finished that work up and moved downtown Toronto and joined the lab of Dr. Joanna Romans. And Dr. Joanna Romans is at Sick Kids at the University of Toronto, and her passion and my passion are to study Shwachman-Diamond syndrome. So this is an inherited bone marrow failure disease that occurs early on, basically within the first year of life is when you would expect a diagnosis. And it's characterized by poor growth, chronic infections due to bone marrow failure, a strikingly increased propensity to develop myeloid leukemias, and a number of other chronic disorders of various organs. And the one that I was particularly interested in was the pancreas. So during my PhD, I basically studied this disease in mice and tried to understand why mutations in the genes that are indicated as being non-functional in this disease, why these mutations lead to a disease phenotype, how that impacts specifically the pancreas, and how that might ultimately promote pancreatic cancer. So that's why I have the pink ribbon in the center of the screen here that I think most of us might recognize as the ribbon for the Cancer Awareness Campaign. And some of you might be asking, what on earth is the connection between all of these things? And that would be the wonderful molecule that is in the bottom left of your screen, the ribosome. So essentially, pokeweed antiviral protein actually renders non-functional the ribosomes of plant cells or any cell that it happens to enter. And Shwachman-Diamond syndrome is caused by variants in a gene that is really important for a proper functioning ribosome. And cancer in general is now known to be a disease that really hinges on the function of ribosomes. What are ribosomes? Well, I don't want to scoop myself too much because we're going to talk about it a bit later, but they are basically, in my opinion, the coolest things ever, and also arguably the most important things, because they are the molecular machine that translate the genetic code into proteins. And proteins are the things that actually do the lion's share of the work in the cell. And so the ribosome is critical because without it, you basically have a blueprint with nobody to read it or act it out. So more on that later, because that also connects to our friends, the viruses, or perhaps foes. Okay, so it's a little bit about me, a little bit about where I'm coming from. And as I mentioned, at the beginning of my research career, viruses were pretty front and center. And then during my PhD, they kind of went into the background, although I'll talk a little bit later about how they actually were still pretty present. And I can tell you that in my teaching, I'm really trying to bring forward the perhaps misunderstood and maligned viruses as really critical components of understanding biology. So what are viruses? Why are they fascinating? Well, they're very, very, very small, which is kind of interesting. They're different from everything else in terms of life. They're genetically tractable, which is a fancy word for saying we can really muck with them and make them do whatever we want. And I don't think I need to convince anybody that they have had a significant impact on humans and in fact our planet as a whole. So to convince you that viruses are interesting, we need to talk a little bit about what they are. So viruses, many have called them genes in a box because they're actually quite simple. And so here in the next couple of slides, I'm going to just take you through a couple of features about viruses. So first off, we're just going to talk about structure. So viruses are surprisingly diverse. There are ridiculous amounts of them. They are virtually everywhere. And they come in a variety of shapes and sizes that are broadly binned into, depending on who you talk to, 4 to 5 categories. So we have our friend, the coronavirus on the left here, and that is one of these round viruses. You have icosahedron viruses, you have rod or filamentous viruses. So here I'm showing tobacco mosaic virus, which was actually a very significant virus agriculturally speaking, but also scientifically speaking, as it was it was the first, I believe, to have its structure crystallized, and therefore a lot of what we know about viruses originated from early work on TMV. We have Ebola, which is kind of a virus that we have in North America an interesting relationship with, because I think our relationship with it is very much Hollywood, whereas significant parts of the world, in West Africa, for example, this is their coronavirus. And it's interesting to think about how we've responded to coronavirus and how we have not responded to Ebola. And then on the right, we have another really famous kind of virus. So bacteriophages are a class of viruses. They have their own variability within that grouping, but they are another really significant virus, both in terms of humans, although I don't think it's quite as commonly known as, say, something like Ebola or coronavirus, but also scientifically, because again, this is one of the so-called model organisms that we use in biology. So again, a lot of what we know about viruses is based on research with bacteriophages. They have been primarily in the spotlight lately in the context of being used in light of antibiotic resistant and multi-drug resistant bacteria. So basically bacteriophages are so named because they eat bacteria. So Greek, phagia is to eat, and so bacteriophage. And so people are using, this has been quite prominent in parts of Asia and only recently catching on in North America, but people have used successfully bacteriophages to help people in the clinic who have intractable disease from bacteria that are multi-drug resistant. Okay, so we've got different structures of viruses here that you can see. Of course, there are more than what I'm just showing you here. And viruses also vary in size, but again, they are generally very tiny, ranging from around 20 to 400 nanometers. So for a bit of reference, 1 nanometer would be 1 billionth of a meter. So they're really, really wee. Admittedly, we have recently discovered some so-called giant viruses, but these are more of an exception than a norm. And generally speaking, we think of viruses as being very, very, very small, to the point that one of the ways that people characterize viruses in the past, and now people will mostly use molecular techniques, was by virtue of whether or not you could filter them out of a solution, because the filters just couldn't get small enough to filter them out. Okay, so that was a little bit about shape and size, but what about constituents? So viruses, again, are genes in a box. So they're essentially made up of a type of nucleic acid, which is where the genes would be housed, and a protein casing. And in some cases, they also contain a lipid envelope. So in this picture here, I have a rendition that I found in the Creative Commons of coronavirus. I believe this is not our current coronavirus, but is instead predicated on SARS of 2003 fame. However, the structures are very similar. So you can see the nucleic acid in this case is RNA, and it is coiled up inside this circular structure, which is essentially a lipid envelope, so fat containing envelope with proteins embedded in it. And the RNA as well is coated with a protein. So this is interesting because if we were to look at a cell, a bacterial cell or a plant cell or a fungal cell or a human cell or whatever you want to think of, you'd have a lot more going on. This is pretty bare bones. Okay, we basically just have the genetic blueprint, in this case RNA, contained within a protein sac, essentially. And in this case, the protein sac also happens to have some fats. So it's quite minimalistic. And this is, again, why it's referred to as genes in a box. The size of the genomes can vary, and the nature of the genomes vary considerably. The diversity is quite remarkable. You can have DNA as the genetic material, or you can have RNA as the genetic material, never both. And then in terms of that DNA or RNA, it can come in different conformations. So our DNA as humans, our genetic makeup is made of DNA, and it is double-stranded in a helix that is linear. Okay, so again, this here is linear. You'll know that the two pieces of the RNA, the two ends are not connected. So in a virus, you can either have linear conformation, you can have circular conformation, you can have double-stranded DNA, you can have single-stranded DNA, you can have double-stranded RNA and single-stranded RNA. And then if you have the single-stranded RNA, there are different permutations of that in terms of whether or not the ribosome translates that directly into a protein, or if we need to go through an intermediate. So there's a lot of different variation, and all of these pieces, size, shape, nature of genetic material, whether or not they have an envelope, these are all the sorts of things that people would use to identify, categorize viruses. With respect to coronavirus, you'll see that it has these really distinctive spike proteins. So these spike proteins are these little trumpet-like, or I don't know if trumpet's the best word, but maybe, yellow protrusions. And these are the proteins that interact with another protein on human cells. And that is how they essentially tether and gain entry to our cells. And they are also the namesake of the coronaviruses. Back in the 1960s, when these were first identified, the researchers that found them thought that these proteins gave that were reminiscent of the corona that is around the sun. And so that is why they are named as such. Okay. So they're small, we covered that. And I also mentioned earlier that they're different. So I tried to emphasize to you how minimalist these viral structures are. I was trying to think of a variety of metaphors. And I actually had some students several years ago now make an argument that viruses were masters at outsourcing. And that is why they're so minimalistic. They just get something else to do all the things that, you know, poor sap normal cells would do. And so I also thought about Marie Kondo, and like, maybe this is like, they're just like really just trying to get rid of everything. Let's get rid of everything. So yeah, so essentially viruses have gotten rid of everything that is arguably necessary for life. And instead they infect cells and make the cells do all the messy work so that they can keep making more copies of themselves. I'm trying really hard to not scoop myself, but it feels necessary to mention at this point that this is part of why viruses are fascinating, because they're the subject of a lot of debate about what it is to be living. And I teach a course called What is Life? And we often in that course use viruses as a sort of foil to think about, well, if the majority, let's say, I think that's fair to say, the majority of biologists think that viruses are not living, they do not count as life, then what does that mean life is? Because it turns out that despite the fact that there are who knows how many card-carrying biologists out there in the world right now, we still can't settle down on a definition of what life is. So viruses are one of the things that sort of irk the evolutionary biologist and the biochemist and anybody who's really interested in these sorts of deep questions, philosophical questions of what is life, because viruses are pretty weird. So to explain this a little bit further, we have to go into molecular biology. So here's Marina's bread and butter, okay? So this is the central dogma of molecular biology. So some of you will have seen this before, some of you may have had nightmares about this, and others will not know at all what I am talking about, and perhaps it's Greek, although I could do that, and that's not what this is. But anyway, so the central dogma of molecular biology was established some like 50 years ago or so. And it essentially is explaining the flow of information from the genetic blueprint to its activation It's raison d'etre. Okay, so generally speaking, the idea is that you have DNA as the inherited genetic material, that's the hard copy, and then that gets transcribed into a slightly different molecule, piecemeal. So not the whole thing, parts of that genetic material get transcribed into an intermediary. And these are genes, okay? So genes will get transcribed into an intermediary called mRNA. And then that in turn gets translated into the so-called action arm of the central dogma, and that would be the protein. So that would be the doers. Now, please bear in mind that I am oversimplifying. There are all kinds of exceptions here, but generally speaking, this is the central dogma, that there is a unidirectional flow of information, that there are two main steps, DNA to RNA, where DNA and RNA are similar in structure in terms of chemistry. And that's why that's called transcription. I like to think of this as basically you have like the ancient text that gets transcribed. Let's go back to Greek. Ancient Greek text that gets transcribed into Greek, modern Greek, and then that gets translated into actions. Okay, so that's the protein. A little bit more detail here, just to keep us all honest. So again, the step between DNA and RNA is called transcription. Here's where we have the genetic code, and we essentially have 4 different building blocks, A, C, G, and T in DNA, and the T becomes U in RNA. And this then gets translated into a completely different language that is made up of 20 building blocks, and those are the amino acids. Here's our friend, the ribosome. Ribosome is the molecular machine that is doing the translating. It basically acts both as the scaffold as well as the enzymatic input to connect together the building blocks that will make the proteins. I have tRNAs in the center here. They're also feeding into the process, and they basically serve as a decoder ring. They act as a go-between, reading the genetic information in the RNA and providing the corresponding amino acid that will be the next building block to build that protein. So a couple of things to point out here that are relevant, especially to our talk on viruses, as well as some of the material that we'll be talking about with respect to immunology. So one is that viruses completely subvert this whole process. First of all, I made a fuss about how this is generally considered a unidirectional flow of information. Well, thank you, viruses, because you taught us about fundamental biology. It turns out that we actually can go RNA to DNA, and one of the most famous and brutal viruses that we know about can do that, and that would be HIV. So HIV is an RNA-based virus, which upon insertion into T cells, basically so-called retrotranscribes into DNA, and that DNA inserts into the human genome where it can be copied throughout the life of that cell. Okay, so that's one piece. The other piece is that, well, hey, viruses don't have ribosomes, so they can't do any of this unless they gain access to a cell. And B, all of these steps require energy. Okay, so that's where on the right, I'm showing you how we've got chemical energy coming in, thermal energy being released. And that's like a gross oversimplification that's basically there to remind me that we need to talk about energy. Because not only do viruses not have ribosomes, which is generally speaking what I always focus on because I'm biased, but as my physiology colleagues would force me to remember, is that they also don't have any genes to support metabolism. And metabolism is critical because it's not like these things just magically do stuff on their own. They do need energy to help push thresholds so that we can break bonds, build bonds, etc., and essentially avoid cells just falling apart. So viruses must gain entry to a cell so that they can take advantage and essentially hijack its machinery so that they can make more copies of themselves. And one of the things that's challenging to think about, and perhaps again heralds that question of what is life, is that as humans, in trying to understand the world, we really try to assign agency to things. We really try to think about like, but why is it doing this? And we really need to think about viruses don't really have agency. And this is part of the definition of, this is part of the reasoning for saying they're not living at all. They're just a bunch of chemicals that happen to, if they manage to get into a cell, interact with a bunch of other chemicals. And hey, if that goes the way it's supposed to go, then essentially we make more copies and more copies and more copies, and they go and infect another cell. So genes in a box. They are basically genes in a box that make more genes in a box, which turns out, again, is a problem for us in many cases, but also really, really quite good in other cases. So one other thing that I want to talk about in terms of this central dogma of molecular biology before we move on, because it'll be important later, and that is that I should have included a arrow or some kind of recycling sign here, because of importance is that yes, this is a more or less a one-way street with some exceptions, but we also need to think about turnover. So essentially all of these proteins get made. So you have a particular part of the DNA, and something happens and the cell's like, oh, I got to make that gene. Okay, so that gene gets transcribed, you get some mRNA, that mRNA gets translated by the ribosome, you get some protein. But whatever elicited that initial response to make copies of that gene and make that protein presumably doesn't last forever. Okay, instead, it's a transient phenomenon, and we don't want the DNA to be forever now turning on that gene and making copies and copies of protein, because A, we probably don't need it, and B, that's going to sap all our resources of amino acids, and then if there's something else that we need, we have no more amino acids with which to build it. So cells are really, really good at recycling. So essentially a protein gets made, and soon after, it gets broken down again. And it gets broken down through a particular process, which I'm not going to go into in any great amount of detail, except to say that part of that process that's really relevant to the immune system is that essentially everything that gets translated in the cell gets degraded, and then some bits of that broken down protein actually get packaged into membranes and put on the edges of proteins that then get stuck on the outside of the cell. So imagine that you have a little hand that's sticking out of the cell, and in it, it's holding a little piece of a broken up protein. And this is the cell's display system. It's basically the window on the shop telling the passerby, which is the immune system usually, what is inside. Okay, this is going to become very relevant when we think about how does the immune system know that a cell has a virus in it. Okay, so the idea is that everything that gets made in this cell gets displayed to some degree on the outside of the cell. And this allows the immune system to say, oh yeah, okay, that's fine. That's that protein that we know about. Or, hey, wait a minute, what is that? Okay, great. So, different from the rest of life. Viruses don't have ribosomes, they don't have a metabolism, they're just genes in a box, okay? This is part of what I think makes them really quite fascinating. So now that we've finished our foray into molecular biology, let's get back to some just basic virus stuff. Some of the words that you might have seen if you have been reading any virusy things are things like the lytic cycle versus a lysogenic cycle. And in general, this is thinking about how do cells gain entry into cells and how do they exit cells. And again, if you remember, I mentioned that the bacteriophage, that's the one that looks like a little spaceship with a little icosahedron on top and the rod, and then the little like landing legs. A lot of what we know about this is based on bacteriophages. And so these words, lytic and lysogenic, usually are referring to them, but in some cases also also apply to other viruses. In a lytic situation, essentially, as we've established, the virus needs to get its genetic blueprint into a cell so that it can make more copies. So it essentially injects the genetic information into the cell. There it gets translated by the ribosome into proteins, and it makes many, many, many more copies of viral particles or virions until literally the cell can't hold them in and it bursts open. It helps that process along the way, but in part it's literally an aspect of capacity. Okay, the lysogenic way of doing things is when the viral genome incorporates into the host genome. So I mentioned how this can happen with HIV, and this can happen with some other viruses as well. And it basically hangs out. And so it goes through what's called a latency period. And in some cases, this means that you are not symptomatic. And this also means that the immune system has a harder time of seeing the presence of that virus because it's not really active. It's kind of just hanging out in genetic form. In fact, yeah, a lot of our genome is riddled with previous hitchhikers that have entered and never left. What about coronavirus? Because that's top of mind for a lot of us. Coronaviruses are enveloped viruses, so they actually have a different mechanism because their envelope, which as I mentioned is made up of lipids and proteins, it actually has the ability to fuse through that interaction with the spike protein and the ACE2 receptor on our cells. It can actually gain entry into our cells without having to go through this specific lytic phase, and it can then actually exit again using similar mechanisms. I've got a video here that's just a little snapshot of cool. So as I mentioned to you, bacteriophages are able to inject their genomes into bacteria, make lots of copies, and then explode them. And people are pursuing this as a treatment for multi-drug resistant bacterial infections. And this is just a cute little time-lapse video of bacteriophage exploding bacteria. So you can see the bacteria sort of swell up, and then they just sadly go, oh, poor little bacteria. So there's some hot footage from the science lab directly to you. This is courtesy of the Howard Hughes Medical Institute, which I strongly recommend as an amazing source of accessible information about science on so many different topics. Okay, now I just need to figure out how to get back to navigating. There we go. Okay, so prolific hitchhikers. So we've talked about how viruses are genes in a box, how because of that, they have to actually gain entry into a cell to do anything. In fact, this is why I know a lot of us have been looking at the news and thinking about how long coronavirus can persist on certain surfaces. And one of the things that I found surprisingly hard to get a straight up answer on is, why do viruses only last a certain amount of time on certain surfaces? So essentially, in part because they're just genes in a box and they don't have a metabolism to help them deal with the environment, which our cells do, they're just going to fall apart, frankly. And if that environment, I'm sure you've heard that if something is in direct sun, then that means that they'll break apart sooner. That's simply for the same reasons that if we sit in the sun, that we can increase our risks of skin cancer. We're talking about UV rays, and these are literally going to be poking holes in the viral particles. So they have to get into cells. If they are not in cells, they're basically dormant. And despite that, they are just everywhere. So they are considered the most abundant organisms on the planet. I use quotes because I can tell that I probably have already offended people in the audience by calling viruses organisms. Sorry. all organisms have evolved strategies to cope with viruses because everything has been infected by viruses. And some of these strategies are actually hot topics in science. So we can think about things like RNAi, which some of you will have heard of, and CRISPR, which I imagine many of you have heard of. CRISPR is actually a mechanism that was discovered in bacteria to help them deal with bacteriophages entering. And so it's a way for them to specifically target viral genomes that are entering their cells and break them apart. And virtually all cells have pretty nifty mechanisms because viruses have been around for a very long time, and essentially all cells have co-evolved with viruses. In fact, that's part of the reason why things like coronavirus are posing such a threat to us, because it's a novel virus, and therefore we as a species haven't had time to co-evolve with it, and therefore sort of balance out the system. Because if you think about it, and here I go ascribing agency to viruses when I know they don't have any, but it's not really in the interest of a virus to kill everything that it meets, because it actually needs cells in which to persist. So if we think about how natural selection works, you can imagine that a virus wants to be able to populate a host, use it to make more, because now there's more viruses, and then do that without basically killing the host. I guess it's kind of like, don't bite the hand that feeds you type of thing. And that's part of the reason why people say that Ebola is kind of like a crappy virus, because it actually kills its hosts, unfortunately, very quickly. And that's part of why it has this, why it doesn't spread as fast as perhaps some other viruses can. Okay, I will skip that other point so we can move on. So another reason why they're fascinating is beyond just like general biology interests and the fact that they are everywhere. They've also had a massive contribution in several different respects. And I just have a couple here. So as I briefly alluded to before, virtually all living organisms have parts of their genomes that once upon a time were part of a viral genome. In humans, that's estimated to be somewhere in the vicinity of 50%. Okay, so think about that for a second. Our genome is about 3 billion base pairs long, so 3 billion building blocks. About 2% of that is actually thought to code for any proteins, thinking back to our central dogma, and about 50% is thought to be viruses that once upon a time entered. Harking back to that epigenetics talk, part of epigenetics in a perhaps less sexy respect is the idea of masking parts of our genome that we don't want to be expressed. So we can imagine that there are large swaths of our genome that are viral that we don't want to be expressed. And so they've been sort of shut down by clouding them with chemical marks. Viruses also recently have been really catching the limelight in terms of an important role in symbiotic relationships. So I'm sure many of us have heard about microbiomes and how cool it is to maybe like take someone else's poop and like put it in you. Ugh, I don't know. But anyway, the microbiome has been catching a lot of attention lately. And there's like some small voices in the corner that are saying, hey, there's also viruses everywhere. So maybe the next hot thing is going to be the virome, because that's a thing. Also, apparently, the ocean is full of viruses, to the point that now people are saying that viruses are contributing so significantly to the carbon cycle that they should really be researched in the context of modulating climate change and ocean acidification. So they're really quite prolific. I've already talked a little bit about how viruses help us think about biology in terms of broad definitions and narrow definitions of what is life. They were also working with viruses was how researchers in the 50s established that DNA was the genetic material. Prior to that, the best thinking minds in the science world would swear up and down that protein was the genetic material. And by that, I mean the inherited material. And it was because of some fancy work with viruses that DNA was established, in fact, as the genetic material a couple of years before the famous reveal of the DNA structure by Franklin and Watson and Crick. And finally, something that's been near and dear to my heart and played a major role in all of the research I have done is that viruses are an amazing tool in genetic engineering. Because they're essentially genes in a box and they're really simple, we can use them to essentially swap out parts of those genes and swap in genes that we would like to swap in, and essentially use them as a template for doing whatever we want to do in the lab. So they're used worldwide in this context. And so this can be anything from generating genomic libraries. So I used viral particles to generate a genomic library to locate the gene and study the expression of the pokeweed antiviral gene. And people can use them to express their favorite gene. They were used in the Human Genome Project because they can also take quite a large segment of DNA into their constructs. And so they've been really useful tools when we're talking larger scale genetic analyses. And they've also been the vectors, the tools that have been used for life-saving gene therapies for inherited diseases and cancer. Admittedly, this has not been all good news. Some of you may be aware that the initial gene therapy trials using, I believe it was adenovirus as the vehicle to carry life-saving therapeutic gene sequence for children with severe immunodeficiency syndromes, it unfortunately caused a severe allergic reaction. And so this is me starting to tiptoe towards the more immunology side of this talk. But essentially, despite some considerable setbacks, we are still seeing how viruses can be used to provide some hope and therapy for individuals who are living with pretty devastating diseases, especially in the context of immunodeficiency. So in the bottom here, I'm just showing a very simple schematic of what this might look like. Where on the left, we have a schematic of a retrovirus, and the idea is that we can replace the genes, and in this case, there's literally just 3 of them. We can replace the genes in the retrovirus with a single gene that will confer a function that for whatever reason is not working well in an individual, and through that way perhaps provide some therapeutic. Okay, and so of course I've talked mostly about good things, but there are also the less good things, and that is disease. And so what I had hoped that we could do, but I have a feeling that we can't in the webinar setup, was to hear from audience members in terms of telling me about perhaps some of their favorite or least liked viruses. But there are several that I can think about and share with you. And to try not to be entirely human focused, we can think about, again, tobacco mosaic virus. And there are many different viruses that have considerable impact on agriculture today. I love cats, and human leukemia virus is also a terrible virus that can really cause awful disease in kitties. And then in humans, we have many, many different kinds of viruses we can think of, including things like influenza. We've mentioned Ebola. There's the viruses that cause chronic illness like HIV and herpes and hepatitis, as opposed to the more severe ones like Ebola and influenza and, of course, coronavirus. But there are a lot of viruses that impact our lives, especially through disease. Some are better known than others. That's one of the things that I like to think about is why that's the case. So let's talk a little bit about COVID-19. I don't have a lot on this because I thought I would save it for questions instead. And I've been trying to do a lot of reading and preparation so that I could hopefully be as knowledgeable as possible for any questions you might have on this topic. But in general, so COVID-19 is the name of the disease. The actual virus is called SARS-CoV-2. Coronaviruses are a relatively new virus in terms of infecting humans. They are, as I've mentioned previously, enveloped, and they are plus strand RNA viruses. So that essentially means that the RNA genome of this virus acts both as the genome and the mRNA. So it doesn't have to do any sort of intermediary step. It can directly translate that genome into protein. Interesting for coronaviruses, a little bit of a claim to fame is that they are the largest known naturally occurring RNA molecules. So their genomes are big. They are the biggest RNA genomes that we know of. Most of us probably are familiar with coronaviruses from SARS. Because I'm from Toronto, SARS really did hit Toronto quite badly, although it was mostly confined to hospitals, but it still really devastated the city. Nothing like what we're experiencing now, but still. MERS was a smaller outbreak, although you can see it had a pretty terrifying death rate. I think it was something in the vicinity of around 2,000 people worldwide that contracted this disease. And this originated in the Middle East, hence MERS, Middle East Respiratory Virus. And then, of course, our current outbreak. You'll see that I've put some question marks after the mortality rate, which would probably be a better way to put that instead of death rate, although I thought to be spot on here. I have some question marks because we really just don't know. This is an estimate, and I imagine that it'll change. I have no idea if it's going to go up or down, but here's hoping for down. Worldwide stats, I checked those just prior to the talk today, and we currently have 14.9 million confirmed cases, 616,000 deaths, which is pretty remarkable. So quite a massive impact on our society. The last time that we had anything even close to this was quite a while ago, and I'm sure many of us have heard about the 1918 flu. The stats are really unclear, but I believe they're thinking somewhere in the vicinity of 100 million infected and 50 million maybe died. So that was quite a remarkable pandemic that really devastated our society. Okay, so don't have a ton of time, but I wanted to talk a little bit about how viruses make us sick and tell you about a couple of things that I think are quite interesting. But essentially, what's interesting is that it's not so much the virus that's making us sick as our response to the virus that's making us sick. So essentially, the virus gets in and it starts to proliferate. It's not until it starts to damage cells that you would really see any symptoms of illness. And in fact, much of what we associate with a viral infection are due to the immune response. So we're talking about things like fever, rash, headache, aches in general. These are all due to our immune response. So a quick crash course in immunity. So there are a lot of different players in our immune system, and we don't have time to go through them, nor am I an expert. But I'm focusing on a few that are relevant specifically for what we're talking about here. So we've got dendritic cells, neutrophil cells, and macrophage cells, and these cartoons are depicting those. Essentially, all of these cells, what they do is that they engulf pathogens. So imagine the virus or bacteria enters your cells, or fungus for that matter, enters your body rather, and these are circulating white blood cells. So they're circulating, and if they encounter a foreign body, they will literally eat it. So it all kind of comes down to just eating things. And so the first line of defense is eat the cells, and the second line of defense is tell other cells about the cells you've eaten. And so the most important cell in that context is the dendritic cell. And so that's the one that's got the star shape over here. These are cells that sort of just hang out all over our body all the time. And then they circulate, and then they migrate back to our lymph nodes, where they sort of show and tell what they've found, if they've found anything. The role in general of innate immunity is to both cause inflammation, so things like that fever and aches and pains and stuff like that. So that's essentially the influx, the quick influx of a lot of white blood cells to a space will cause things like pain, as well as they cause fever through this interesting interaction with the brain, where you actually see constriction of blood vessels in extremities to prevent just random heat loss. And so you're basically trying to keep the heat in. And you also actually send signals to your cells to up the metabolic rate. So remember how I talked about how we need energy to do all these cellular processes? Essentially, a fever is trying to ramp up metabolic processes, create a hotter environment, because it'll help degrade the genetic components of the virus. And so the innate immunity and the inflammatory response is an early response that is trying to sort of do a quick and dirty and generally non-specific way of keeping the disease at bay, while some cells, namely the dendritic cells, move to the lymph and then essentially report to other cells that, hey, I saw this thing, you should maybe come check it out. So that's where the adaptive immunity comes in. So those dendritic cells basically go to the lymph and they literally present, they're called antigen presenting cells. Antigen is a fancy science word for thing, and they present the foreign thing to the lymphocyte cells. And so we've got two different kinds. You've got T cells and B cells, which are named based on their origin. T cells come from the thymus, B cells come from the bone marrow. And essentially, the dendritic cell says, hey, T cell, check this out. And the T cell has its own receptor. If the T cell's receptor matches the thing, the antigen that the dendritic cell has found, then it's like, what the hell? Because it normally just circulates around holding out this hand that wants to touch things. And because it's been trained to not react to your normal cellular doings, remember how we're displaying stuff that's in the cell on the surface. So the T cells go around, they're like, yep, that's normal, that's normal. And then the dendritic cell comes along, or maybe the T cell sees on another cell and is like, what the heck is that? And that essentially triggers the alarm. The alarm essentially means that that individual T cell, each T cell has its own specificity, will now activate and proliferate and go through what's called a massive clonal expansion, where it'll make ridiculous amounts. We're talking millions of copies of itself within a couple of days. And now those T cells can flood your system, identifying more cells that perhaps, more antigens, so more pathogens that have that little thing that it picked up as no-no, and mark them for destruction. The B cells basically do a very similar thing. They also have a little hand holding out, looking for something to bind to. And once again, if they basically can match their hand and to the T cell hand that's been activated, then that's an indication that, like, yep, we need to go to town. And that B cell will now, again, same deal, activate, make ridiculous amounts of copies of itself, but it also makes antibodies. And then it'll secrete those antibodies out into your system, and they will basically circulate around and find things to bind to. And that essentially tags things for destruction. So we can think about our immune system as essentially heating stuff up to melt the virus, tagging virus and whatever else is like pathogens, other kinds, for destruction. And one of the things that we have to think about is, we all know that fevers are really, actually kind of dangerous. And it's interesting to consider that they're evolutionary conserved in all vertebrates. And we know that they improve outcomes with infection to the point where there's some evidence where drugs have been given to reduce fever and they've actually seen a poorer outcome for the patient, but there's a trade-off there. We have to be very careful because yes, we see a boosted immune response for some really interesting reasons. I don't have time, but if anybody wants to ask, I'm happy to share. We see a boost in the immune response with higher temperatures, but at temperatures higher than 41 degrees, we actually start to see cells just dying, like our cells start to die. So that's quite dangerous. And finally, interferon. So interferon is another sort of cool protein that gets secreted by our immune cells. And it does a lot of neat things and is actually being looked at right now as a possible therapy for coronavirus and for COVID-19. Because essentially what interferon does is it's a protein that gets released upon stimulation by viruses. So essentially viral RNA is detected, the cell's like, oh dear, what is this? And it starts to secrete an interferon. Every cell that sees that interferon through interactions between that protein and proteins on the membranes of those cells will start to do a variety of things that will essentially shut down the protein translation system. Remember, viruses don't have ribosomes. That's the whole reason they're there. So it'll shut those down. It'll cause the cells to die. Viruses actually are really good at making cells not die so that they can keep living in certain cases. So it does a few different things that are pretty nifty, all of which essentially leads to the virus not persisting. So of course, viruses have evolved proteins that directly interfere with interferon. And so one of the therapies that people are pursuing is to give synthetic interferon to people that have severe infections to sort of swamp out any antagonistic ability that the virus might have by just giving a ton of interferon, and that will then hopefully help them shed the disease. Okay, so essentially we're coming to the close here. Just a couple more things. So viruses, I've hopefully convinced you, are pretty cool and also pretty much everywhere, and also kind of terrible in certain contexts. And our immune systems are really good at dealing with viruses, believe it or not. I realize that I don't want to be insensitive because I know that many people have been impacted by this disease terribly, especially our senior populations. However, I do want to just point out that our immune systems are amazing. And if we have the right tools and time and we live in healthy spaces, then they can really do a lot to help us from disease. So I'll just close by telling you one very short, cool thing about viruses, cancer, and our immune system. So essentially, I talked to you about how cells present stuff on the outside to tell the immune system what's inside. And that's one of the ways that our immune system can detect viral infections. It turns out that this is also a way that our immune system can detect cancer, because cancer is a disease that is caused by changes in the genome, okay? There's a massive amount of change that happens in the genome before cancer can really happen. Because there's changes in the genome, that means there's changes in mRNA, which means there's changes in protein, and that means that what gets displayed on cancer cells changes. So this is how our immune system can detect cancer cells, because their display system is showing something different from the regular cell. On top of that, it turns out that cancer cells are really quite a welcoming home for viruses, because cancer cells, to get to the point where they're actually able to be cancerous, have shut down a lot of the checkpoints and security features, shall we say, that cells contain to help them deal with problems like cancer and like viruses. So a cancer cell is kind of like a safe haven for a virus. These things come together in some exciting new therapeutic avenues for cancer. So many of us, probably all of us, have had loved ones or have ourselves dealt with a cancer diagnosis and are well aware of how well, barbaric a lot of cancer therapy is. If we think about things like chemotherapy and radiation therapy, these operate under a scorched earth approach of basically poisoning our bodies in the hopes that the highly metabolic cancer cells will get more poison first, die faster, and that our healthy cells will recover. That's generally the principle here. However, we now realize through some interesting work at the turn of last century by a unethical scientist who realized that his patient that had cancer and was basically terminal got a influenza infection, or I can't remember if it was influenza, but they got a viral infection and suddenly were better. They were like, wait a minute. And essentially what can happen is that if you have something to trigger your immune system to sort of wake it up and be like, hey, pay attention to what's going on over here, that it can come in and actually clear out the cancer. And so people now are using these principles that originated from this idea of this weird, unethical doctor who then went ahead and tested cancer patients by infecting them with things and things that terrible things. But anyway, so that happened. And now we're using that to do things called immunotherapy, which are basically helping to boost the immune system. And one of the ways that this is being done in this fancy picture here that's from the scientist is they're basically taking out cells from patients with severe cancers like dendritic cells and other antigen presenting cells. So these are cells that are going to go show things to the T cells and be like, hey, T cells, stuff's happening. And they basically take them out of the patient and show them the thing that they should show the T cell, and then put them back in the patient so that they will show the T cell and mount the response. So that's one thing they're doing that's pretty cool. The other thing that's even more sort of esoteric and wild is that they are basically using viruses to engineer T cells to contain the tag already. So they're taking T cells out of patients, they're re-engineering them using viruses, using that swap out technology that I alluded to create the antigen presenting receptor that they need so that they can now go and target the cells that are cancerous and kill them. And the very last example that I don't think is depicted in here is that they're using something called oncolytic viruses. So these are viruses that you train to specifically infect cancer cells. So now one of the things that's beautiful about all this is that we don't have the scorched earth approach of just blasting every cell. We have targeted approaches here. And in this case, you're basically engineering a virus that will target cancer cells, infect them, make many copies, and then burst them using that lytic phase that I talked about earlier. That bursting now not only kills the cancer cell, but it also essentially barfs cancer antigen all over that area so that the immune system will wake up and be like, oh, hey, something's going on here, come in, clear it out. And these therapies are showing a lot of promise. Again, there's always, another shoe to drop. There's also some major problems, but nevertheless, I think this is one of the biggest hopes that we have right now for having some cutting edge therapy. Okay, so that's all I have. I'm sorry for going a little bit over time, but hopefully through this quick talk, I have managed to convince you that viruses are indeed fascinating for a variety of reasons, but probably most importantly, because they have a significant impact on humans and our planet. And so it's just interesting and important for all of us to learn a little bit more about these tiny little things that some of us call organisms. So with that, I'll say thank you for your attention, and I would be happy to answer any questions. And for anybody who is seeing this on their own time, or that we don't have questions that can be answered right now, please feel free to reach out at me by email, and I have my email address there. Thank you.
Thank you, Marina. And I figure we can leave that slide up there for now, just in case folks want to copy down your email address. We don't have any audience questions that have rolled in yet. Folks, please do feel free to comment in the chat box on Zoom or on the Facebook chat box, but that means that I can get all of my questions answered.
Sure.
Because when I have a biologist captive like this, I simply have to pick their brain. So let me dive in. One of my questions was about your slide on COVID-19.
Mhm.
And you said that the coronavirus contains the largest RNA molecules ever detected. I'm wondering if that has an impact on their virulence.
Mm. I don't think so. I think the main thing that's making them a particular challenge right now is just this straight up novelty. Um, you could imagine that more genome means more possibility to mutate, but in some ways, part of the reason that viruses are so pernicious, I guess, is the fact that they are small and compact and therefore, I guess, nimble in that respect. So to my knowledge, I don't believe that has an impact on virulence.
Very good. Jeanette's next question. Okay, so I wanted to ask, and I know you said at the beginning of the talk you're not a virologist, so please feel free to defer to someone with more expertise. But another sort of virus story that has been in the news was that in the last couple of years there have been 2 patients that have been cured of HIV. The Berlin patient and the London patient. And I don't know much about that, aside from the fact that I believe at least one of them received stem cell therapy. Do you have a layperson's explanation for that by any chance?
I'm hesitant because I definitely remember reading about this, and now I'm worried that I won't remember things correctly. I know it's quite controversial, and that so I guess what I do feel comfortable saying is I believe the controversy is about simply that because it's such a small number of individuals that have had this work out, is that it then makes it difficult to rule out other variables that might have contributed, but nevertheless it is quite remarkable. And I feel like it's a really interesting story too, because if I remember correctly, I think at least one of them has been quite involved himself in terms of promoting this. But yeah, I'm not sure. Sorry.
That's okay. That's why I gave the caveat at the beginning.
Yes, thank you. I appreciate that.
We have a question here in the chat box from one of our attendees asking, do you think the coronavirus will mutate to become less lethal? Is there any chance of that?
Oh, I mean, I think there's just as good a chance of that as anything else, really. I think, again, one of the things that we have to keep holding on to here is that there's no agency here, and that it's more about selection and survival. So if we think about that, we can imagine that mutations are happening randomly. And for those of you who are interested, there's an amazing website where you can actually watch the mutations. It's called Next Strain. It's all one word, but I highly recommend checking it out. So the mutations are happening randomly. And then the question is, how is selection going to operate on those mutations? Given that we generally would think that a virus would survive better were it to be less problematic for its hosts, then we could speculate that might indeed happen. But we don't know. But it is an important thing to bring up, because I do think that there was a lot of fear mongering in the earlier days, in terms of a lot of my God, it's mutating and blah blah blah, and it was kind of well, yeah, it's going to do that. Part of why RNA viruses are so tricky to work with is because they use a kind of polymerase. So this is the enzyme that's going to make copies of the genome. So when they're basically replicating themselves, it doesn't have a proofreading function. And so our DNA polymerase that replicates our genome is pretty darn good at doing that in a manner where it won't incur any errors, because that's what's worked for us. We are, if you think about it, the complete opposite end of minimalist. And so there's so many moving parts that we can imagine it's therefore fairly simple that a slight change would cause a big problem because it's so like tightly wound together. Whereas with the virus, it's like, sure, whatever. It's much more versatile in that regard. And because of that, you could argue that there's been no selection pressure to have a better replicating machinery. So we're gonna have lots of replication. That's totally normal. Mutations should not be scary per se. And we could maybe speculate that over time, we would expect it to become more virulent. So we can certainly hope.
The same person in the chat said, did you see the report that said that 1/4 of people in Delhi show immunity?
I didn't. I did see that in the last few days, there's been quite a lot of reporting about cases in India, because it went from like, why is India's cases so low? And then like all the counterpoints of like, here's why and blah, blah, blah. But I hadn't seen that. That's actually quite interesting. I don't know. I'm going to have to look into it.
You really blew my mind with this whole viruses. Are they living or not? I feel like I'm going to lie awake tonight thinking about that, because I went back to my like undergrad biology and again, and I was like, well, they're not a single-celled organism. They're not bacteria. They don't have any of those mechanics that would allow a cell to replicate. What the heck do you call this thing? Like, is there a name for like something that's halfway between living and inanimate? Like, it's wild. I've never thought of that.
It is wild, and it's interesting to think about if virologists are housed, of course, in like life science and biology departments, and I wonder if they have moments of existential angst in terms of that. But yeah, absolutely. I mean, our general conception of life is that it is cellular, that it has cells. And viruses, by our current definition, do not have cells. However, there is a large problem in the definition in that we only have one example of life, and that is the life that we know on Earth. And so there are people who speculate that we should be less rigid. And a lot of the definitions that we see for life it quickly becomes evident that it's hard to say cut and dry, no, viruses don't belong, because it's weird to say that something is not a thing because it shares some of these characteristics, but it doesn't have a bunch of these other ones. And then you can actually think about how that's problematic in our own daily lives in terms of some of the prejudice that we can see in our society and be like, well, wait a minute, why are we making these choices? But in biology, we have to make these choices. We operate under classification systems, but I think when it comes to viruses, have to operate with a little bit of nimbleness and just keep our minds open. Because, for example, one of the common things is to say about viruses not being alive is that they can't persist on their own. But you can quickly come up with everything from like fairly practical arguments, if we think about symbionts and things like lichen, for example, to more philosophical arguments of like, but who is truly able to survive alone? And therefore be like, well, I don't know that that's a good thing. But
and that's where we have to get Darcy in.
Yeah, precisely.
To give us the philosophical angle.
Precisely. But yeah, I will though take a quick opportunity to do a PSA and just point out that it's important for people to realize that viruses are not the same, because part of our multi-drug resistant bacteria problem is because of people taking antibiotics when they shouldn't. And the mechanism of action for many antibiotics is for them to actually disrupt the ribosomes of bacterial cells. And viruses infecting us are not infecting bacterial cells. And so dismantling the ribosome of a bacterial cell isn't going to help you. And B, viruses don't have ribosomes. So it really is neither here nor there. And then there's other antibiotics that attack cell wall structures, which again, the viruses don't have. So next time somebody tells you that, oh, I'm going to just take an antibiotic anyway, tell them that that's useless and in fact part of the problem.
That's a great PSA. I wanted to come back to a question that you sort of planted during the talk, which was you talked about a boosted immune response
Mm.
during fevers. And my like undergrad biology brain thought, well, maybe like thermal energy is energy, and with more energy, you can create more antigens more quickly.
No, it's a good
That's
idea.
my guess.
So from what I can gather, based on like some excitement, I think it was last year or something like that, where there was this like quick byline in the science news about people figuring out why fevers are good. It's not fully understood, which is interesting in itself, because like I mentioned, this is something that is conserved in all vertebrates. And there's all this evidence collecting to suggest that they are good, and yet we're not quite clear. And if we just go down to basic chemistry and physics, we know that if we heat things up that are living too much, that there are problems. Like we can just think about how an egg changes its color, and that's basically the proteins in the egg losing their structure. And so when we have a fever, the dangerous part is that all of the structures in our cells that are doing important things are, living by the same laws of physics that the virus is. And so it's problematic that we're heating up and therefore essentially loosening everything. But it would seem that at least one of the clues that came out, again, I think it was last year, was a major part of our immune surveillance is these T cells are essentially moving along our vasculature, trafficking and recirculating to try and surveil our body. And it would seem that they did some really elegant work and showed that at certain temperatures that are in the febrile range, that the trafficking of these cells was improved and changed. And this was mediated by a really famous so-called heat shock protein. So the thinking is that they're essentially in part boosting the immune response by making everything move a little faster. So that's kind of neat.
That'll be a really fascinating, I don't know, bit of business to keep an eye on, like, yeah, it's fascinating. Well, I have one more question on my list, and we're going to wrap up at 8:30, folks. So last chance if anybody in the audience wants to ask a question, but Jeanette's going to ask her last question now. And it's that I have been, thinking a lot about the idea of spillover. David Quammen's great book called Spillover talks about how most of our worst viruses came from animals. And of course, so that's part of this discussion around coronavirus. And I was wondering if kind of explain that concept to us. I don't, again, for the layperson, whether you can explain why so many viruses might come from animals, jump from animals to humans. What can you speak to that?
Yeah, so I was actually, so this is something that's really interesting. So the fancy term is zoonoses. It's got like a weird spelling, right? And this is the idea of any kind of disease that comes from animals. And it turns out that, this is pretty common. There was a really nice article, I'm just looking at some of my notes here, in The Scientist about this as well. But it's not 100% understood. There was some interesting stuff about bats, and I think in general they've been talking about bats and I believe pangolins. I don't even know if that's how you say that. But it seems like, so with bats, there were some really weird ideas about how bats are like a repository for viruses. And I guess the question there is like, why aren't they getting sick? And there was a couple of ideas, some of which were like molecular and, you're like, oh, okay, like there's slightly different proteins and therefore slightly different bindings. So maybe it's not as efficient. So some of that was like cool for someone like me. And then there was other stuff that was just like wild, which was basically bats fly. And to fly, they have a really high metabolic rate. And so one of the ideas is that simply that high metabolic rate acts like a fever and helps them fight off infection. So that was like a random, okay, who would have thunk that? And then the other thing that I wanted to just add on that topic was, I know there are concerns. I was reading about the, I don't remember which, I want to say H5N, and I can't remember which one it was, but it was an influenza that has been in general monitored this is in China. And this is the one where they're basically going around, and every time they see it in chickens, they go and they kill all the chickens. The concern there is that essentially they figured out that the virus in that context is only essentially one mutation away from being able to infect humans. So in that context, the birds are getting sick. The question is whether or not they can pass it on to humans. And so in that context, the idea is that, well, we better basically eradicate any animals, sorry for the animal ethics there, that contain this virus, because if it gets into another human and has the opportunity to replicate in the human, the natural selection, the selection pressure in that context would be that any chance mutation, and again, mutation is pretty common in these viruses, that would change that one piece to now allow it to effectively infect humans would be devastating. And so it seems like there are two cases. There are the zoonoses that actually also make the animal sick, but haven't quite crossed over to humans, but maybe are close in that in the chicken case. And then there are the other cases where they don't make the animal sick, for example, with the bats. And that might be through some slightly different genetic makeup in the bats that changes interactions between things like, say, a spike protein and their receptor versus some more physiological and behavioral elements that might be allowing that organism to not get infected by this thing.
The pangolin is such a random addition to that list.
It's funny because the Squamish Library has the which is delightful, the program where they give books to newborns. And so we had a baby girl a couple of years ago now, and the book that we got was an African alphabet, and in it had pangolin. And I remember being like, I've never even heard of this, and now it's like pangolin, pangolin, pangolin. So yeah, thank you, Squamish Library, for alerting me to the existence of pangolins.
That is a great book, by the way. I have perused that board book myself. But yeah, it's fascinating to think like, you know, that there would be enough human pangolin contact to even discover that there was a zoonotic virus there. But of course, I'm just looking at it from a Canadian worldview. Maybe some people spend every day with a pangolin.
It's entirely possible. And it also brings up the other dimension that a lot of people, so one of the sort of depressing things about reviewing all this literature for COVID and that I've been doing over the last couple of weeks, I'm hoping that I can incorporate this into my teaching in the fall too, has been, it's depressing to see how many articles in both the literature as well as like news media, et cetera, in the past, say, decade have had alarm bells, like pretty strong alarm bells that we are not ready for a pandemic, it's coming, all this stuff. And then you're like, oh, that's unfortunate. And one of the things is this link that people are really pointing out now about how the climate change and changes in land use policy are going to make these things happen even more frequently as we push people closer and closer together, as we have climate refugees again being pushed and pushed closer together. So there's some interesting and in a depressing kind of way information about how and why we can expect to see more pandemics, unfortunately.
I think that was sort of the conclusion of David Quammen's book as well, which came out like maybe 10 years ago now. And he said the same thing. You know, it's not an if, it's a when. And it's, you know, he couldn't pinpoint what animal we were going to see it cross over from, but Yeah, he mentioned the inevitability of it. Great book, not very lighthearted, folks, but really, really well written, popular science book if you're into that kind of thing.
Mhm.
Well, it is 8:30, and so it is time for us to say goodbye for the night. I want to thank you again, Marina, for being our speaker tonight. Um, and for those of you who are listening out there in the audience, this is the end of our sort of summer quest series. But hopefully we'll be back with more virtual talks soon. For those of you who are into this format of talk, we are going to be hosting a discussion on August 20th with Dr. Rudy Reimer, who is an Indigenous archaeologist from SFU, and he's also a member of the Squamish Nation. So we haven't received his exact topic and abstract yet, but we're really excited to have Dr. Reimer speak, and that will also be a partnered event between the libraries. So stay tuned for details on that. The date is definitely August 20th, so you can mark your calendar and stay tuned for more details. Thanks again to the other libraries. Thanks again to Quest, and thank you, Marina.
You're very welcome. It was fun.
It was fun for me too. I love having all my questions answered. And of course, folks, this talk is being recorded, so you will be able to find it on Facebook after the fact. We're going to say goodbye to Facebook now. So goodbye, everybody on Facebook.